So now it is just 2 p.m. We will start our today's program. Good afternoon to everyone. Today is the third day of our national workshop on climate smart livestock production in India. Today we have 2 sessions and a quick Valedictory Program at the end of today's session. In our first session, session number 6 of the workshop, we will discuss on impact of heat stress on livestock production and health. It will be discussed by Dr. Vishi Das. And the next session is the 7th session or the second session of this today's program. We will discuss on genetic adaptability and resilient livestock production under environmental stress. The speaker is Dr. S.M. Dev. So, before the cutting of our scientific session, first there is one important announcement that today is the last day of the program. So, we decided that the presence of today and yesterday is very essential for getting your certificate. So, we ignored the feedback of the first day due to some problems in some connectivity and other regions. So that's why it is very we request to all the participants, please fill up the feedback form of the last 2 days, yesterday and today's program. And then after we will discuss details about how do you get the link of the overall or final feedback form. That we will discuss later. Now we will start our today's program. First session is on the impact of heat stress on livestock production and health. I am requesting Dr. Joydeep Mukherjee, Assistant Professor of the Department of Veterinary Physiology, and Dr. Deepak Banerjee, Assistant Professor of the Department of Veterinary Physiology, to act as the rapporteur of both the 2 sessions of today's program. And now we will start our first session. The speaker is Dr. Vikas Chandra Das. Dr. Vikas Chandra Das graduated in veterinary science from Vidhan Chandra Krishi Vishwavidyalaya in 1991, and his master's is from National Dairy Research Institute, Karna, and doctorate in veterinary physiology from West Bengal University of Animal and Fishery Sciences, Kolkata. He worked as assistant professor at Vidar Veterinary College for a brief period of time and joined ICAR as scientist in 1997 at National Research Center on York. Later, he shifted to Indian Veterinary Research Institute, Bareilly, and worked in December 2019. Now he works as principal scientist at National Research Center on Pig, Guwahati. He did his postdoctoral research from University of Cologne, Germany, and on return he worked on stem cell at IVRI Bareilly. Right now he is working in the field of stress physiology. He has guided around 11 postgraduate students and published 35 research articles in peer-reviewed journals. With this brief introduction, I am requesting Dr. Vishi Das to deliver his speech on impact of heat stress on livestock production. Dr. Das, please start.
Thank you, Dr. P.K. Das, for a nice introductions. And as being introduced, right now I am at National Research Center on Pig, Guwahati. And recently I have been shifted from Bareilly to Guwahati NRC Pig. Madan sir, namaste sir. And then this is a topic which is given to me by the organizers, to speak on this. This is impact of heat stress on livestock production and health. And this is a very pertinent one in today's context, when the global warming is very evident. And every one of us, some or other ways in life, we are facing the stress. Of course, if we ask that what is stress, there is no clear definition as yet, because the etiology is not well defined, the prognosis is also not well defined. And as you know, the stress, it has become a part of life, and it is not inherently bad sometimes. And all life forms, we have evolved some mechanism to cope with the stress just time to time. Coming to the stress, as we speak, because it comes from different environmental stimuli, there are different psychological stimuli, and because of that, we feel the strain, and thereby we get the just effect or the response. The individual, as an individual, when he perceives the threats to our homeostasis, then that become the real stress. The stress is sometimes we call the stressors, and when the stress response, they truly threaten the animal well-being. The animal experiences the distress. So the non-threatening stress, which we can call it as a good stress sometimes, and when the stresses it exerts the deleterious effects on welfare of the animal or the human beings, then we can call it as a bad stress, or it is also called the distress. Coming to the next, that is called the it is a kind of model of animal stress which is given by a scientist Moberg in 1985. Therein he stated the three different stages of stress. Where in the first one he says the recognition of a stressor. Our system has to recognize what kind of stress it is, and thereby it exerts the biological defense against the stresses. And after all, at the end of the day, we see the consequences of the stress response. It is the last stage, what we call the consequences of stress. That is the very important, and that determines whether his animal is suffering from the distress or merely experiencing a brief episode in its life, and that will have no significant impact on its welfare. So, as we see the consequences at the end of the stage 3, that if the animal fails to cope up with the different stressors, thereby it comes to a pre-pathological state, and it develops the different pathologies which is evident from the effect of the stress. Then coming to stress response, here the scientist Moberg, he has given a beautiful diagram to express just to detail what is the stress response. As you can see, there are three different bar diagrams wherein the different functions have been stated: F1, F2, F3, and Fn, like that. And this diagram is particularly called the non-threatening stress, and means this no distress, and it is this period, the brief period of distress, and the biological resources what is available, and that is being used for the biological functions. The resources what is available in the body, that is being used for the biological functions, and the biological resources is diverted until the resources. If you see the first diagram, the available for the F1, that also remains same in the last stage when the animal cope up with this stress very well. That means the recovery stress and the stress period both are equal. Now, if you come to the next one, wherein you can see the stressors, they have become little bit more, and thereby the animal or the individual, they will experience the greater recovery period. That means the biological resources necessary to cope up with the reserve of the stress, and it impairs the biological functions leading to the stress, and it requires the much longer recovery period. So you see here the recovery period between the second and the third one, the recovery period is much higher. That means your stress is very high. Now the scientist Moberg, he has given one more diagram wherein he says that when the one stress is already there, and when one other stress is exerted upon it, that means there is a synergistic effect of the stress 1 and the stress 2. So in that case, some of the functions of the animal or the individual, it is being compromised. So that means there will be deleterious effect on the body response. Then you see there are several stresses. As you could see, the metabolic stress will be there, poor housing. So just because of poor housing, there may be overcrowding. Just because of transportations, the social distancing, and of course there is heat stress is always there because of high temperature, high ambient temperatures. And animal will thrive and try to cope up all those stresses, but at the point when all these things there will be a break point, and animal will succumb to the pathology and even to the death also. Then environmental stress, and as you know, it all depends on the intensity and as well the just duration of the stress also. So in the first step, the animal will try to cope up with the animal response. That there will be behavior changes, there will be just autonomic nervous systems, the endocrine systems, and the immunological endpoints. So all these factors, they will come together. They will try to cope up with the intensity and duration of the environmental stress, and animal will go with the normal functions. But when the threshold level is crossed, obviously there will be impaired functions. And the function is the impaired means there will be reduced performance on the health and the productions. Coming to the climate and the weather, when it has got direct effects and the indirect effects. When there is direct effects, we could see the quantity of the feed available resources will be reduced. The quality of the feed also will be degraded. And the heat stress, severe heat stress will be there. And obviously there will be water scarcity will be there. And at the end, the livestock will face the disease consequences. Pathological state will be very high. And there may be a lot of disease vectors also would be available in the environment. And the indirect effect, animals will try to adapt certainly, and there will be microbial populations will be very high, and host will try to develop the resistance to the different infectious diseases. Then these effects on the bioclimatic stress on the animal productions. As we know, there will be different physiological functions. Those who are being changed due to environmental stressors. There will be heat loss mechanism will be very much active. Neuroendocrine system also will be active. Mineral balance there will be active, and the water balance and quantity of the feed also may be reduced. Then, as we all know, that where in the thermoneutral zone, if the temperature goes down too low and which crosses the lower temperature, the point will come when this metabolism will be stopped, and because of hypothermia, animal may succumb to the death. The other way, when it crosses the upper critical temperature, animal may experience hyperthermia. There is the animal will try to cope up with this. Lot of operation rate will be increased, metabolic rate will be high, and at the rate, if it fails to cope up, then animal will succumb to the death. Then this is a funnel model where the partitioning of the dietary energy is given. So intake energy, digestible energy, metabolical energy. As you could see, some of the energy could be there will be waste due to material waste. And the energy which is available at the end, we call it the retaining energy, and they will be helpful for the fattening and the growth. And there will be external products. Some of the retained energy could be used for the milk, egg, offspring, and the wool productions. And of course, the heat loss will be there due to basal metabolic intensity, activity of obtaining nutrients, and combating the external stresses. So big problem is that metabolism or the heat productions. Say one adult human being, the heat produced is almost equivalent to 100 watt bulb. And in case of the lactating dairy cow, it is almost equivalent to 1600 watt bulb. So there must be some avenues of the heat loss. This is the animal when it is exposed to ambient temperature, and when the sunlight is the main source of the energy. So these are the different sources where the animal gets the energy. And of course, the animal has to evaporate the heat through the perspirations and the sweating. The infrared thermal radiation from the animal will be there, and the wind also will help to evaporate some of the energy. And this conductive and radiative energy requires the temperature gradient to happen. All these events to happen, we require to have a temperature gradient. Otherwise, these are the mechanism will not work certainly. And this effective as the air temperature approaches the skin temperature. So it is certain that when the air temperature approaches the skin temperature, obviously these methods of conduction, convection, and radiation would be less effective. There are many different levels of sensitivity, like take the breed, gender, health, age, geographic location. Yes, it's very important, and the acclimation durations. Then, as you could see, the use stress, which are the good stress, and there will be the optimum productions. But when the temperature goes high, the performance will be equally compromised, and the critical zone will come, and wherein the animal stop producing there will be no productions. The animal may succumb to the hyperthermia. There will be weight loss, the dysfunction of different physiological functions, and the animal may succumb to the different diseases. And the distress, this kind of distress could be as the stress would be very high, and this could be taken as a distressed, and the animal may succumb to the death. Then the assessment of thermal comfort, as call it as a discomfort also, and which is stated or denoted by the temperature humidity index. And here it has been postulated that by the THI less than 72, that means animals is in comfort zone. And if it exceeds 88, then animal is facing the severe stress. And there will be different physiological responses like respiration rate, pulse rate, ectal temperature, all will go high, and the feed intake certainly it will go down. And in case of the lactating animals, if this temperature is 25 around, then there will be some depression of the appetite. If it is around 30, there will be marked decline in the feed intake. And if it is 40, virtually the animals stop eating. And in case of the avian species, the 1 degree rise in the temperature beyond 25, 1.5% of the feed intake would be reduced. Then this is one diagram which shows that what do you call this? In case of the but this is to show the effect of heat load on the meal kit. And in this, during thermoneutral conditions, if we say -1 to -7, the first diagram if you see, there the temperature meal kit, the meal kit will be very high and will be very optimum. But whereas the temperature just during the heat stress, just during day 1 to 140, during day 1 to 14, if you see, the meal kit will be drastically reduced. Then depression in the feed intake, as it could happen during the summer stress, there will be lower DMI, and because the hot weather, it reduces the nutrient availability, and obviously there will be, so as the absorptions and absorbed nutrients also used very less efficiently. And how this physiological mechanism it operates during heat stress conditions, this may lead to the ruminal acidosis. How it happens? When the animal is in stress, obviously respiratory rate will be too high. When the respiratory rate is high, the more and more carbon dioxide from the lungs will be eliminated, and the decreased carbon dioxide level will be there in the blood. Once the carbon dioxide level is decreased, that means blood pH becomes more alkaline, and this reduces the bicarbonate concentration in the saliva, and which is indirectly increasing the risk of ruminal acidosis. So it is very evident in case of the summer stress. And this is the effect of metabolism. And during the heat stress, the ruminant liver they become functional with regard to hepatic glucose output. And the glucose is preferentially utilized for the process other than milk synthesis. And the hepatic glucose output remains increased during the increased glyconeogenesis and also increase the gluconeogenesis. So these are the events which really get adapted during the carbohydrate metabolism. And during heat stress, just now I said that hepatic glucose is used for the process other than milk synthesis, alteration of the glucose turnover, and increased glucose demand for the energy need. And we know the galactose does not enter in the mammary epithelium directly. So the 2 molecules of the glucose is needed for each molecule of the lactose. So there will be lower availability of the glucose for mammary gland lactose synthesis. And in fat metabolism, yes, the NEFA levels are typically reduced in heat stress animal, and heat stress animals have the higher insulin level, and which we know that it is the lipolytic hormone. And coming to the protein metabolisms, the protein just synthesizing machinery is just muscle protein synthesizing machinery, and RNA and DNA synthesis capacity are reduced, and similar effects are apparently occur with regard to mammary alpha and beta casein synthesis. And as the skeletal muscle catabolism is clearly increased during heat stress, so it is evident that a lot of plasma markers they will go higher, they will be expressed during muscle breakdown in variety of species. And coming to heat stress on the productions, we know that there will be heat stress. The marked evidence is that this estrus activity is also just lowered in case of the heat stress. And the silent ovulations could be observed, which we call the silent heat, and ultimately which may lead to the poor embryonic implantations and increased embryo mortality. And this is a schematic diagram which shows that when there is a heat stress, this how the uterine environment it gets affected, because there will be the there will be decreased feed intake, and the animal may experience the negative energy balance, and indirectly which may lead to the less output of the GnRH and the LH, and there will be decreased estradiol secretions, poor estrus expressions, and the prolonged follicular dominance will be there, and the low fertilization rate, poor embryonic, and which may lead to the embryonic loss. And this is the same phenomena where the summer high ambient temperature, the heat stroke, how it the female infertility and the male infertility. And this heat stress challenges the reproductive performance of the male through a variety of altered physiological means. And this already I have told you, the altered follicular development will be changed, there will be lower estrus activity, the embryonic development will be there. So these are the different stages, how it could physiological phenomena it is expressing this. And if we talk of the fertilization rate directly in the lactating cows, which are exposed or not exposed to the heat stress, it has been published that it has been published by several workers that in non-heat stress animal in lactating dairy cows which are facing this stress but which are not threatening to the physiological system, in that case the fertilization rate would be as high as 88%. However, the animal which is facing the distress or the stress which is threatening to the homeostatic system, so that the animal that the fertilization rate may be reduced to as low as 55%. And this is the heat stress which leads to male infertility as the result of apoptosis and the low sperm count and the germ cell DNA damage and prevention of oxidative damage, cell survival and sperm with the DNA damage, which may lead to the male infertility. And coming to the livestock health, there will be, because of high temperature, there could be vector population may be increased, and this is just changes in the rainfall pattern also may support to the vector during the wetter years. And beyond vector-borne diseases, helminth infestations will be greatly influenced by the changes in temperature and the humidity, and climate could also influence the disease distributions. And associated with the environmental conditions favoring the intermediate snail host, and outbreaks, if we talk of the anthrax during the outbreaks often associated with the altering heavy rainfall, drought and the high temperature. So these are different axes like HPA, like hypothalamo-pituitary adrenal axis, then SAM and thyroid axis. These are the different axes which may lead to the negative impact on innate immunity, cell-mediated immunity, and humoral immunity. However, some other pathways, the metabolic axis, somatotropic axis, and other hormones, which may really stimulate these different defense mechanisms like innate immunity, hormonal, and the humoral and the cell-mediated immunity. That means the negative impact and the positive impact both are evident through this diagram. And glucocorticoids, as we know, they inhibit the aspects of immune functions, and as it causes decrease in cytokine productions, and the antibody production is also decreased, and the proliferation of the immune cell is also decreased, and thereby the leukocyte subset population, there will be a lot of changes, and the glucocorticoids inhibit the aspect of functions. The review of the heats. Now, what I have talked so far, just will have a short review on this. And coming to thermal stress, core body temperature, yes, it will be high. Respiration rate, it goes high. Skin temperature will be high. Blood flow and sweat rate will be very high. And thereby, so as the blood flow to the internal organ, yes, it will be reduced. Salivation, drooling, and panting rates, it will increase. And the heat production rates and metabolic rate also will be decreased. And heat loss via radiant, conductive, and convective avenues will be decreased. And at the end, the animal would suffer from the dehydrations. Immune status, there will be immune functions will be compromised. Susceptibility to parasitic and non-parasitic diseases will be high. Mastitis will be evident. Somatic cell count will go high. Plasma antibody level would go high. And at the end of the day, animal may succumb to the death. Nutritional status, the DMI intake, yes, it decreased. Energy requirements of maintenance, it goes high. Salivation, it goes high because the saliva to the rumen, it comes down, and salivary carbonate pool to the bicarbonate pool to the ruminal buffering system, mechanism, it fails. Ruminal pH, it goes down. Animal experience the acidosis. Potassium loss from the skin goes high. Dietary requirement of potassium and sodium, it goes high, and water intake also it increases. Milk production, it is down. Mammary blood, yes, obviously it will be down. Mammogenesis, it is down. Lactation peaks, it is down, and milk component level, all it goes down. Endocrine, hormone linked to the metabolism, all hormone like thyroxine, somatotropin level, it comes down. Hormones linked to the water and electrolyte metabolism, antidiuretic, ADAs or aldosterone, it increases. Catecholamines, epinephrine and norepinephrine, there increases. Prolactin level, it goes high. Prolactin receptor numbers, there are decreases. Leptin, yes, it increases. And it has got the effect on insulin, which goes high. And suddenly the blood glucose level, it comes down. Reproductive status, breeding efficiency, it is compromised. Conception rate also it decreased. Fetal and postnatal mortalities, it increases. Calf birth weight, it decreases. Semen quality overall, yes, it decreases because the spermatogenesis is hampered. Sperm motility reduced to a great extent. Percent abnormal and aged spermatozoa, it is also increases. Estrus activity, the duration is decreased. Head detection, it fails. And uterine blood flow, placental weight and the growth is down. In a retained placenta, the incidence of the retained placenta, it increases. Gestation period also, it is reduced, and the labor and difficulties also it is experienced. Reproductive status, follicular development will be very low. Oocyte quality, it is decreased. Multiple ovulations, it is enhanced, and corporal luteal size also decreased. Thank you very much for being with me. Thank you.
Thank you, Dr. Das, for your nice presentation. You have details explained the physiological mechanism of the stress and how it affects the different systems of the body, of the particularly relationship with the production. Thank you. Now I am requesting our rapporteur, Dr. J. Mukherjee, please place some questions for discussions.
Thank you, Dr. Das, for your nice and lucid presentation. I got two questions from the participants. Number one, whether any individual could claim to remain stress-free.
Yes, this is very difficult to answer, but Madan sir is seeing Dr. Madan itself is a severe stress is now imposed on me. And sir is there, those two stalwarts. In fact, I tell you, there is no reason being stressed when Dr. Madan is there. But yes, I feel stressed when Madan sir is there. See, now, coming to this question, if any individual it claims that he is stress-free, I really do not subscribe these views, because it has been seen that our saint, or the kind of people, those who had no psychological distress or any kind of the disturbance from their family life or anything, but even then it has been seen that this hits of protein, if it is taken as a marker, it has been seen that around 2 to 3% or 1 to 3% of the HSP still it is expressed in case of the people, those who claim that they are not in stress. That means the stress is evident with every individual, even though somebody claims that I am not in stress, but yes, some amount of stress, it may be psychological stress, or it may be in some other form, which because the threat perception itself is a very just difficult kind of thing, because that it is a threat to me, that may be different to the others, because you may not take that similar threat as a stress to you. So the perception of the threat is very important. So in that way, every individual in throughout their life, always they'll have some sort of stress in their life, even if it is claims also there is no stress, because it is by taking this HSP expression studies and others, it has been seen that yes, there will be some sort of stress always there. Thank you.
Okay, now, next question is that, is it justified that any stress markers or indicator could be equated with different animal species?
No, I could not get it. Can you repeat this, please?
Okay. Is it justified that any stress marker or indicator could be equated with different animal species?
Well, see, there are, as of now, there are several stress indicator or the markers as has been worked upon. Like there will be several transcription factors, HSP, these, that, lot many isomers will be there, and then physiological response, then hormones, corticoids, cortisol, and lot many the markers will be there. Suppose, but yes, this it is with emphasis. We cannot say that, as I told you earlier, that one trait, one kind of stimuli, which is a threat to one individual, that may not be a threat to the others. That means, so the indicator, what are the indicators as of now it is available? They are not the real just litmus test for all these stresses. what I mean to say, I just give you one example that in case of the in a critical environmental conditions when the experiment is conducted, in case of the stallion, one example I am giving you, in all other animal, when the cortisol is taken as an indicator or the stress factor, but at the same time, when the stallion is exercised, when the stallion is allowed to mate with the mares, because the mating with the mares is a pleasure to the stallion. That cannot be a stress. But it has been seen that during mating, the stallion, the corticoids level, it goes high. So the same indicator cannot be equated to all the animals, all the individuals. So the indicators must be different for the different individuals.
Okay, thank you, Dr. Das.
Okay, thank you. I think we will have to complete our sessions in time. So if there is no question, very thanks to Dr. Mukherjee also. Sir, Modan sir is there, Sir Kamra sir is there. Sir, do you supplement anything, sir? Modan sir? Modan sir?
Is mute, unmute.
Sir, unmute and you can tell something, sir.
Message him.
Modan sir, you unmute, sir, first. Kamra sir, can you talk something, anything about this lecture? Kamra sir?
Yeah.
Yes.
He has taken a part of nutrition also, nutrition as well as the physiological stresses. So naturally, it is individual how much tolerance every animal has. He is correct in saying that if same stress is given to different types of animals, naturally their response will vary a little. Not much, but there will be some changes. There will be some differences between these effect of stress and different types of animals. I agree with you. It's a very nice presentation by Dr. Das. Thank you.
Modan sir.
Modan sir, can you supplement anything? Modan sir? Sir, would you want to tell something, sir? Probably some problem is coming.
Sir, it's not, system is not okay over.
No, actually, sir has posted some message in the chat box that it is a good presentation for a workshop. The student training should be benefited by the basic facts. The stages of adaptation need to be explained for building the concept. Thank you, sir.
Sir को कुछ बोलने के लिए बोलो.
Okay. So I think today we can conclude this session, first session of this program. Thank you, Dr. Das, for your nice deliberations and discussions pertaining to the questions which already placed by Dr. Mukherjee. So again, from the organizers, I would like to thank again Dr. Rishi Das for your nice presentation and discussion, the physiological basis of the stress and livestock production systems, how it is related to this climate smart resilience. So, okay, many thanks to Dr. Das. Thank you. So now we will start our second session. That is the last session of
Dr. Das, is
yes, sir.
Dr. Babu in there?
No, sir. Today, Dr. Babu, he has not joined.
Korea?
And no, sir. Today he has not yet joined. He may join. Now
I have received a very sad news from Bhubaneswar. And I just wanted to confirm from him that what happened. This is with respect to Dr. Kageshwar Pradhan, our former Vice Chancellor and eminent nutritionist, physiologist, nutritionist, Dr. K. Pradhan. I just was wondering if
He's no more, sir.
detailed
no more.
confirmation.
He is no more, sir. I also received the message.
Yes.
He is no more.
Yes, that's what I have heard, and that's very sad.
Yeah, yeah.
What happened?
I also-
He
There
was
was
not
some
of corona.
heart problem.
Details we did not get.
I think in case that Guha is there, that you can just
Okay, sir, I will just.
maybe at the end today,
Yes.
that it gets confirmed through Dr. Guha
Okay.
that in this workshop that you do say a word or so relate to Dr. right?
Okay, sir. Okay.
How to listen to Dr. Dev.
Yes, and now we will start our seventh session, the last session of our 3 days national workshop. Our speaker is Dr. Shitangshu Mohandev. Dr. Shitangshu Mohandev has joined Agriculture and Research Service during January 1992. During his scientific career of 28 years, has served in different ICR institutes at different capacities as head of the campus, head of division, and director, etc. He has vast experiences in working in different agroclimatic zones and studied many species of livestock like cattle, buffalo, crow, sheep, camel, yak, legume, etc. He has successfully completed 15 research projects, has published nearly 100 research papers, 4 books, and guided 13 PhD students. All his students are highly successful in their career. Under his leadership, the first test tube calf of ER was born during 2013. He is member of different committees of national importance. He is a fellow of NIHEC, IAVR, and is a senior executive fellow of Harvard University, US. To this very brief introduction, I request Dr. S.N. Dev to talk the workshop on genetic adaptability and resilient livestock production under environmental stress. Dr. Dev.
Thank you, Professor Pradeep Das. Good afternoon, everybody. My esteemed teacher, outstanding scientist and academician, Professor M.L. Madan, sir. Professor Sagar Khanal, my esteemed senior. Dr. D.N. Kamra. I could see Dr. J.R. Khan, Dr. Sejian, Professor Pradeep Kumar Das, who is organizing this program. Friend Dr. Prabal Ghosh, Dr. Vikas Das, dear participant of this workshop, ladies and gentlemen. I am the last speaker, so it has certain advantage as well as disadvantage also. Disadvantage is that something will be repeated. I think you will have to bear with me. Advantage, I feel some of the question asked by this participant, possibly that will help me to deliver it in a better way. The physiological aspect to the stress has been explained, as well as what is the animal is contributing to the environmental stress has also been explained by earlier speaker. So I'll be explaining on this genetic adaptability and resilient livestock production under environmental stress condition. If we see just briefly, the type of stress due to environment is primarily, I classify the water stress, housing stress, feed stress, disease stress, and thermal stress. So out of these 5 categories, I can classify these housing, feed, disease. These are related to, again, heat stress or thermal stress. Water is also related to thermal stress. I explain in this way, water will be there, but potability of the water for our livestock rearing or human consumption may be reduced, but total water will go nowhere. So if you see this just briefly, you bear with me because some are repetition. Thermal stress is a complex phenomenon which challenges the homeostatic mechanism causing alternation to normal physiological function. In 2014, it has been worked out by a group. 1.8 million tons of milk is lost in our country, which is around 2% of the total milk production due to thermal stress. And 2015, this ADB, Asian Development Bank, they have estimated 8.7% reduction in GDP by 2100. It's a serious. And from biodiversity angle, FAO has estimated that if the temperature goes from 1.5 to 2.5% enhanced, there will be 20 to 30% animal species will be extincted. If you see this particular diagram, this is global temperature increase rate. In 120 years, it is around 0.7% enhancement has occurred. But there are certain countries where it was colder area, but if you see the Indian context, within the 50 years, 0.5% increment in temperature means temperature has been observed. So within 100 years, we will be having the 1 degree increase in temperature, means 150 years from now, there will be loss of 20 to 30% of the animal species. And this bottom, these are some intensity. Incidentally, it is the southern, western, southern, and northern parts is mostly affected. And the frequency also, this eastern part, they suffer less. It is again western, southern, and northeastern region. I would like to draw here this particular diagram. Possibly it has been explained. The temperature humidity index, it increases, heat dissipation also increases. Body temperature, animal try to maintain up to this threshold THI. Cow tries to maintain the homeostasis. But what will happen if the THI increases further? Rectal temperature will increase, animal will not be able to maintain the body temperature, and there is an increased risk of mortality. I think Dr. Das also explained. So what animal does at that time to cope up with, it reduces milk production. Also, it reduces metabolic heat production. This is the mechanism animals tries to sustain when it crosses the threshold of THI crosses and goes further. So heat tolerance, I would like to explain here, and it is well established. This is balance. Animal try to ability of the animal to balance the thermogenesis and heat dissipation under temperature above thermoneutral zone. And resilience is the capacity of the animal to be minimally affected by heat stress or to rapidly return to the state part in before the exposure. Again, I am coming this particular area. As a breeder, our interest is this milk, fertility, and this life of this animal. It should not die. So these are the areas. So the question was asked, I think I will be trying to relate this thing also. We have here, yes, there is a species-specific advantage and disadvantage in our country. This biodiversity, one hand, we have yak, there's a species, it can sustain minus 50 degrees centigrade. Another hand, we have camel, which can sustain at 50 degrees centigrade. So whatever the situation, we'll have species. Sometimes there are reports that cow in some of the African countries, cow is replaced by goat to accommodate in a better way under the thermal stress condition. If we see this particular to sahiwal and frijoal animal, there is a study at CIRC that sahiwal, they produces differentially express this heat shock protein 19, which helps them to adapt in the climatic high temperature condition. Again, if you see this photograph, Harperker cattle, this bull in this region, you see this is a dark coloration, which during summer, it goes away, this coat color. This coat color, it changes, and this is also controlled by genes. Subsequently, I am coming in my slides. So, as my topic is related to genetic adaptability or selecting animals genetically which are superior, who can sustain better in a high temperature or humidity condition. So we partition it as a breeder into two components. One is a non-genetic component and another genetic component. What is happening? The respiration rate goes high, skin temperature increases, rectal temperature increases, feed intake also it decreases. So this can be addressed by altering the cow's environment or nutritional management, like adequate shade, cooling, adequate water, modify ration, or change in feeding time and frequency, and some feed supplementation. But the genetic component, what I am interested to deliver here, that is it is the adaptation, is the ability by low metabolic rate or increased heat loss of an organism to survive under heat stress condition. Genetic variation among animals, it is observed within a breed or between breed, it has been observed. So we try to harvest this portion, and there is a methodology, we call it genetic selection for heat tolerant animal. There is a conventional as well as genomic methodology. I will be coming subsequently. And the point I want to emphasize here that the genetic improvement is a permanent and cumulative changes. I would like to draw the paper here, environmental stress as an evolutionary force. I think first day we were discussing, I was listening, is a Darwin's theory of evolution. Yes, it is happening. The animals which can sustain better in the under a high temperature, high humidity condition, they will be naturally selected. And subsequently, this is the basis of species formation or speciation, Darwin's theory. But it takes millions years, but human requirement is faster, so we'll have to take the advantage of it in the micro environment. This period, what is changes is occurring within a group. In a large population, we need to select those animals so that they can survive and perform better. So here my point is physiologically heat tolerant animal, we define the one animal who maintains homeothermy under high heat stress condition. But for a breeder or breeding point of view, we don't limit there. What we say, maintaining productive and reproductive level under heat stress condition is the animal we are searching for. Whether the animal is maintaining and doing well and reducing the reproduction does not serve our purpose. Here in this photograph, this is a study in US. So if you see that there are three groups of animals, this is a high producing animal, this is average producer, and this is bottom is low producer Holstein cows. It has been observed when the temperature increases, that is daily average temperature increases in their country, it is with their respect to the high yielding animal suffers most, and this medium performer, they suffer less, and the low performer or bottom producer, they suffer less. So I'm coming to this selection methodology as a breeder, this conventional selection methodology, just this I think you all know it. We first collect the phenotype of the animal with respect to any trait. Here, my concern is heat tolerance is the trait. So it is composed of, one is genotype, another is environment. And genotype, again, it is partitioned into one is additive genetic variance, and we call it dominance or say non-additive genetic variance. And we use these two methodology primarily. First one is the least square method, wherein we put any value, say this is a milk yield or heat tolerance attribute, whatever may be, there will be mean value and all non-genetic factor their contribution, and lastly there will be error component. This is the least square methodology. Subsequently, there is a Henderson, they have developed another block model or mixed model or animal model. Principally, it is the breeding value of the animal is a factor multiplied by the heritability times. This is the formula we use. And heritability is nothing but the phenotypic variation, what proportion of the phenotypic variation is due to additive genetic variance, which we want to harvest. And sire constant means superiority of these sires based on the daughter's performance over the population or other sire or population. But here issue the coming in, this maintaining productive and reproductive level under heat stress. Here challenge is how to measure this heat this tolerance phenotype. It is a very challenging issue because several major people have tried. Rectal temperature is one, respiration rate, heart rate, sweating rate, hair shedding rate, body surface to mass ratio, body blood parameter. And out of these, it has been observed that respiration rate has a higher heritability, means phenotypic variation is a numerator and denominator is that additive genetic variance. So 76% of the variability is due to genetic origin. So it can be used. Some people have used it. And mostly genetic evaluation of heat treatment or tolerance have been used as a performance recording under heat stress condition. This is the best way of selecting animal for heat tolerance ability. And THI on the day of performance recording is assumed as the heat load affecting the productivity. And this, I think, diagram, I think it has been explained. So it is the heat stress threshold, then middle, and this is a moderate. I think Dr. Das also told when 80% the humidity it increases, and temperature, even if low temperature also, at 27 degrees centigrade with 81% humidity, it also causes severe stress to the animal. So those are conventional. In our country, we are still following that conventional methodology, but heat tolerant animal we are not searching for. We are still trying to find out this milk production and trying to increase it. But molecular breeding or marker assisted selection, this is developing elsewhere in developed countries, mostly US, Australia, Europe. Here the basic principle is that polymorphism in candidate gene, means genes and its protein, how it is contributing to the trait concerned. Any variation, we call it primarily SNP, single nucleotide polymorphism. Whether it is inside the gene or in the vicinity of the gene can be used as a marker, and it can be associated with the traits. Since 1987 to 2004, people were working on it. For example, the heat stop protein in 90 gene, I already told in my earlier slide. Here is a new finding I think I would like to share you. Sleek hair gene, it is on chromosome 20, is responsible for producing a very short and sleek hair coat. First, it was found in Senep pole cattle and introduced in the Holstein Friesian to improve their heat tolerance ability. Because this US is more concerned that in the global warming and other thing, because these Holstein, these animals were the most sufferer. So they tried to introduce, and this Florida University, they have developed two bulls. And this is one of the bull, which can able to regulate the rectal temperature, less pronounced reduction in milking under heat stress condition, and they are selling the semen, and there is a huge taker because in the US, everybody is bothered about that global warming. I think they have the more acute losses they are facing. So this is one of the best things they are doing at this moment. If we see our country, our Jebu cattle or Indian cattle, they have certain advantage, adaptability because of their higher density in sweat glands, and the gland is larger in size, so sweating rate is higher, so they can better dissipate the heat. Also, these glands are closer to the surface of the skin and has more layer of cells in the epithelial layer, so quickly they can transfer metabolic heat to the skin due to less tissue resistance. Another one is, I was telling the Tharparkar cattle skin color. There is a study, this MC1R and PMEL genes, these two genes are highly expressed during winter than summer season in Tharparkar cattle. So it gives a dark coloration, and in summer its production is very less. So level of their expression altered with the heat stress related parameters. So presently, as I reviewed, that 431 gene, including heat shock protein gene, have been found to be affected by heat stress, and approximately 20% of this gene are also contribute or affected during cold stress. Here, there are two types of differences has been observed. One is specific expression of the gene because of the polymorphism, even if it has been observed, the temporal sequence expression also, with time-dependent changes is also observed in this all this group of genes. So I'm listing quickly. I would like to sharing with you. Heat-shock protein gene, there are 58 gene has been identified. These are the list. And these are non-heat-shock protein genes. This red one is down-regulated, and this black, this font, these are up-regulated. So here also, this is down-regulated and up-regulated. 58 non-heat-shock protein genes. So these 116 genes, they have been found to be substantially contributing to the heat tolerance ability of the animal. But after 2004, why I was telling today is 2020, after 2004, people were involved in this elsewhere, not in our country, for selecting animals. But the problem they faced, the candidate gene approach, there are a large number of candidate genes contributing to the ability. It's not only heat stress, is not a single phenomenon or group of gene or group of, I think physiologically we have seen it or it has been explained. A lot of physiological activities are involved. So a lot of genes certainly involved, and roping in all this variation in the individual gene or candidate gene will be difficult. Therefore, this gentleman in 2001 from Australia, he has suggested another selection strategy, that is a genomic selection. What we can do, there was genome-wide association study of those SNPs instead of taking individual marker. It's all these marker, maybe in even thousands or even lakhs of this SNP. There is a methodology I will be coming. So genomic selection, they have tried. And what is it? I'm trying to link it. Accuracy of breeding value using conventional breeding method, though used in different countries, always remains questionable because of its accuracy. But presently, no better option available in our country. That also we should admit and accept. The cost of recording of phenotype in conventional selection is very high. In genomic selection, genetic effects are estimated for each marker and then summed up to predict the expected breeding value. In conventional method, we use the data, phenotypic data, partition it, and find out what is the expected breeding value. Here, directly, this marker effect is added to produce the breeding value. So we call it genomic expected breeding value from all associated DNA markers spanning entire genome. So we are not bothering about the individual or candidate gene, it's the whole genome we are talking. And there are a lot of advantage. One is certainly genetic gain will be improved and its reliability will also be improved. So principle is like this. First, we in a population, I have explained in another slide in a better way, just this principle I'm telling. Phenotype has to be recorded in a reference population, which is a part of a larger population. We also should have it, it is called SNP chip or microarray. So we can get in a single go lot of SNP available in the genome of a particular animal. Based on these two phenotype as well as these SNP data, we develop a prediction equation. Once the equation has been established, we can use it by taking any animal sample. We can, in a single go, we can go for this, we call it SNP screening or chip microarray, and we can predict the genomic breeding value of the animal. So here advantage is that in this, when we will be using this prediction equation, we don't need to evaluate or record the phenotype of the animals once, but this prediction equation is required for the reference population, a part of it. So here the principle of this Illumina is a joint. They have developed a lot of this DNA chip. Initially, they started with 54,000 DNA chip. Then subsequently, it became to 777,000 SNP, and it is increasing up to 90,000. What happens? These are called chip. If it is a complementary, it will hybridize and give a signal. Taking those signal, we can say SNP is available or not. So if you see, this is not a very small task. Manually, it's not possible. There are different software available. We can just, even if 7,077 means 7,77,000 S&P in a single go, putting in a single experiment, even if in a chip 96 sample can be given. So it is a huge data is be generated, which can be churned and produce this equation. And the prediction, once I was telling this is the same thing in a pictorial form. So once we develop that equation, then subsequently we can use it as a for the genomic estimation of breeding value or genomic expected breeding value. But here then question may come, when they have developed, why we are not using in our country? The problem lies here. The equation is population specific, means the reference population and the population where the prediction equation will be utilized should be drawn, reference population should be drawn from the same population. Otherwise, this prediction equation may not hold good in our situation. So we'll have to develop our own prediction, and Government of India recently has put a lot of money, NDDB, NBHR, NDRI, and BAFE, we are working on it to find out a prediction equation so that we can develop a chip and the genomic breeding value can be estimated very quickly. So I'm drawing your attention to this particular, thing. If any developmental organization, because I was associated with this Punjab government, they purchased semen from this Holstein. So this is very interesting thing. You see these days, they are mentioning in their sire catalog the genomic breeding value. Without this, no country is purchasing the sires or semen of the sires available in US, Europe, or say Australia. So genomic breeding value is, if you want to sell it, our semen, or use it, the genomic breeding value, possibly people are concerned nowadays. So what is happening? And this is commercially available genotyping service, Genes Diffusion. It is Europe-based. France, Belgium, then Poland, Netherlands, Spain, US, even in China is taking advantage of it. What it does, it offers breeder a hard genomic evaluation. You can take this hair follicle from any animal, send them, they will give the genomic breeding value of this animal. What they are doing in the chip, what I told you, that microchip or SNP chip, they have put 40% this variation with respect to production, 15% with respect to other health, 20% with respect to fertility, and remaining 10% is type, and hoof health is very important in case of Holstein. So it is 12.5%, and milking speed also 2.5%. Why it is important? Because we know in Indian condition, when we are trying to improve the milk production, fertility is going down. They are inversely related. But this methodology, you can say 40% weightage, 20% on fertility, other health, it is taking care of all the economic component of the production, livestock production system. So farmer will be more benefited without compromising the overall productivity, health, and other related issues. But here one caution is that the first line, if you see in 2017, that Australia first, they have evaluated genomic expected breeding value with respect to heat tolerance. But here they are claiming SDSNP chip, 61% accuracy. So my dear, this participant, you can very well ask this question. You are telling accuracy should be very high. Why 61%? Where this 40% or 39% has gone? Because science is developing. What we find in this process, if you see this particular diagram, there is an epigenetic modification has come into play. From a single gene, due to RNA processing or RNA alternate splicing, one immature RNA may produce 3 different types of RNA, mRNA, and finally 3 different proteins. In other way, from one gene, we could have 3 different proteins based on some other factors. So ultimately, this protein is contributing to the physiological or metabolism of this animal. So whatever gene is available, we are targeting the gene, but we find the protein level also there is a variability is coming from a single gene. Similarly, I would like to draw your attention on this particular. Here is the genotype space, first one. There is a new terminology I find, it's internal phenotype and external phenotype. For example, let me give, suppose if we say gene A, B, C is contributing to the fertility of the animal. But fertility is the external phenotype. Internal phenotype is whether it is contributing, it is helping this differential expression of LH or not, or some other hormone. So those are the internal phenotype. What is happening in the internal situation, we are unable to measure. But externality, because of this LH or this particular oxytocin or these hormonal changes, the phenotype varies. So people are thinking D level, they have decided, they are trying to find out the internal phenotype. They are putting a gadgets in the milking machine itself. And on day-to-day LH, the concentration is also recorded along with the milk yield per day basis. So having said this thing, we will have to, the science is progressing, and because of this epigenetic modification, it may happen without the genomic selection or genomic expected breeding value, possibly after some time down the line, we'll have to go for proteomic or metabolomic genomic breeding value or say breeding value estimation in future. So, but there is a silver lining I would like to share here. A question was asked, which species will win and lose in a warmer climate? It depends where they are evolved. They have done an extensive study, though it is in rainbow trout. They have taken rainbow trout from different climatic zone and found out what is the effect on the heat in this species of animal and in this fish. Based on that, they postulated winners are those evolved in warm and subtropical region. Those evolved in temperature ecosystem are the loser, risk becoming extinct. So this is a silver lining for our animal, the Zebu cattle. We have advantage, but disadvantage is productivity is low. So what we should do? First line, if you see huge gap in productivity, that Holstein or Jersey or Brown is whatever you say, it is selected for higher milk yield over the years. And Zebu, by default, it is adapted against heat tolerance. This animal, as I explained, that evolution and the stress, environmental stress theory, how it is contributed to evolution. Maybe in a smaller scale of time, these animals, some of them are heat tolerant, tolerant, and it has emerged or adapted. So suggestion for the animals evolved in cooler ecosystem or in our country where extensive system of rearing cattle or other animals are there, in that case, we have two choices. One is selection of heat tolerant within this group when farm resources, that is adequate feeding, management, heat mitigation strategy, animal health are not limiting their survival. We can even think for this selection with heat tolerant ability within this group can also be one of the strategy if other factors are abundant there. Crossing with Zebu, because this concern with the people who are in the temperate zone, but it can be applicable in our system also. Crossing with Zebu and selection for production, reproduction, and heat tolerant in if farm resources and the health environment means that parasitic pathogen are insufficient. But in our situation, when animal evolve in a tropical ecosystem, we have two choices. One is selection for higher productivity, reproduction, also heat tolerance, because already it is adapted for a long production goal if we have. But if we require a immediate increase in production, because crossbreeding in our country came in these 70s, because of this second region, we required a huge quantity of milk, and this traditional or conventional selection methodology without crossing, it was not possible to provide. Today we are giving 340 grams milk per capita availability. It could not have happened. So that time it was a requirement people have different opinion. There is a debate whether crossbreeding was correct or not, but it was required at that time. Only if we see the availability, it was only 80 grams. Today is 340 grams availability. So I think in the last census also crossbreed number has increased by around 30%. So we cannot simply wish away the crossbreeds. So it will be there. So production, this in the crossbreeds, production, reproduction, as well as heat tolerance, if demand for milk is to be managed immediately. With this word, I thank you and I end my lecture here. Thank you.
Thank you, Dr. Dev, for your nice presentation. You have nicely explained the role of various genotypic factors from the issue of the heat stress, particularly in different situations in our Indian context. So you have nicely explained the marker-assisted selection of highly adaptive animals, particularly our native charm class. So we are very much thankful to you for your nice deliberation and detailed explanation from your genetics and breeding point of view. Now I am requesting Dr. Deepak Banerjee and Dr. Jaydeep Mukherjee also to place the questions for discussions. Dr. Deepak Banerjee. Yes. Unmute. Dr. Banerjee, unmute yourself. Yes.
The first question is to you that is in India, huge number of animals are non-restricted, but they are well adapted in their home site. Please light on whether any steps are taken to isolate such type of genetic material which are responsible for their adaptation.
Can you repeat? I could not very clearly hear you.
Sir, actually the question is regarding the huge number of animals in India are non-restricted, but they are well adapted in their home site. So please enlighten us later whether any steps are taken to isolate type of which are present in the animal which helps helping their adaptation.
I'm again sorry, Dr. Pradeep Das, Professor Pradeep Das. Maybe here.
Yes. Dr. Banerjee, you please try to hold the microphone to your mouth.
Can I see the chat?
Yes, just now you tell again. Dr. Banerjee, you please repeat the question.
Sir, am I audible, sir?
Yes, now it is audible. Please tell.
Slowly you tell.
Yes, sir.
Tell.
Tell.
Hi. Yes, sir.
Yes.
So the question is that in case of indigenous, in India, so many non-descriptive breeds are there, which are having the genetic material which are helping them to adapt that particular climatic condition in India. Is there any steps taken by the government or by the any institute to enlighten us about this type of genetic material which is helping them to adapt this particular type of environment?
Okay. Yeah, another question. I will take both the question, then I will respond. You have another question?
You can place the second question, Dr. Banerjee.
Second question is regarding the cold stress, not only the heat stress.
Okay.
Sir, am I audible, sir?
Yes.
Am I audible, sir?
Yes.
Sir, second question is regarding the cold stress.
Okay, I got it.
Is there any relationship between this in case of the genetic material which is responsible for the heat stress, those genes are also responsible for their adaptation in cold stress? Is there any relationship or different genes are there for that different types of the stress?
Okay, thank you. So first question is whoever asked this audience, from the audience, this is a question, it's a burning If he's a breeder, I can very well understand this question. If each and every breeder has this question in mind, what will be happening with nondescript animals in our country? 10 minutes, Dr. Das.
Yes, please.
There are 10 minutes left, so I want to elaborate.
Yes, you can explain. You can explain.
Okay. So nondescript animals, we are having 150 million nondescript or say indigenous animal. Out of 150 million, only 4 million has been described in breed like Saiwal, Red Shindi, Tharparkar, Kankrej. So 146 million are those animals. The question is around that figure, 146 million animals, they are certainly adapted under the harsh climatic condition, not only harsh climatic condition, harsh nutritional condition also. Those animals are thriving well. They have that ability. Disease condition, they can survive under hard condition. Those attributes we did not see it earlier. That's true. But people in Western world or developed world, they are trying to find out there is a gold mine. So they want to put this germplasm back to their country. I will not be surprised if Holstein is crossed with those nondescript animal in future to introduce this adaptive ability of these animals. So this is going to happen, I can assure you, down the line after 15 or 20 years. Certainly it will happen. Government of India plan is that out of 150 million, only 4 million, which is described by NBG AGR, there is a nodal agency. So mostly Gir, Tharparkar, Shaiwal, and Rathi, we have a program. That is, we call it All India Network Program, All India Coordinated Research Program. CIRC, Central Institute for Research on Cattle, this is a nodal agency. They have a program. They are trying to rope in mostly farm animals because those are in the government farm or the university or ICR farm. They are trying to coordinate research funding is there. Primarily, it is Shaiwal, Gir, Tharparkar, and I think Kankrej also they are taking. So 146 million animal, nobody is talking or thinking as on date. But I find this animal is living on zero input system. Because if you see Eastern India, then Northeast India and parts of the Central India, the input, there is no fodder. They are on the grazing land, on the rail line, side of the rail line, highway, they are grazing. Or after harvesting of paddy or something like this, they have been allowed to graze. So they are reared on zero input system, whatever maybe they are giving, even if cow dung that is helpful for the fertilizer. So these animals will stay there. Government doesn't have, we don't see any plan here, but it will be a gold mine down the line. Again, I am repeating, it will be happening that it will be asked by those countries who are rearing Holstein, Brown Swiss or Jersey animal to introduce these genes into their population. This is the first question answer. Second is cold stress and heat stress. I told in my presentation also, 116 gene I listed, 20 genes has been found to be contributing or differentially expressed in cold stress. Okay. And cold stress in livestock, very less studies has been carried out. It is in, I think, rainbow trout, there are a lot of work has been done. Certainly, these are the genes which is contributing. In addition to that, there will be difference. Suppose I work on yak, with the cold stress means if the temperature at minus 20, animals are most happy. But it is a cold stress for say, our animal who are in the plain. But for yak, it is comfortable because their physiology and other this skin structure will be different. Certainly, if cold stress is a factor, and it is not happening in our country because the area people have invested a lot of money in research in cold stress area, but out of this 116, adaptability has been found to be related to cold stress also. Thank you. Any question? I
Sir,
am.
sir,
Yes.
next question. Can you hear me,
Yes.
sir?
Yes,
Sir,
please.
another question: which is the best breed of cattle for milk production adapted to climatic condition of eastern India?
Okay. Best breed of cattle.
For milk production in which will be adapted in eastern India.
Okay, I understand your point. But thing is that no, to answer this question, uh, breeder always study population. If 5 or 10 animals is doing well, we may not be impressed with that. If the whole population is performing, there is no such study has been carried out. Okay. But Saiwal, it has been observed, pan India, their performance is better. So Saiwal cattle can serve better under Indian situation, West Bengal or Eastern region also. This is the assumption. There is no, I would say, concrete study to establish my concept. But based on the data elsewhere, Saiwal is used in Northern Plain, Southern Plain, Western Plain, they are doing well. So with this, in Eastern Plain also, Saiwan can do well. But Gir has been observed. Maybe this sort of a minister has been impressed with the Brazilian Gir. So he told that Gir should perform anywhere, but it is not happening. Gir outside Gujarat, they are not doing good. So Gir population in Gujarat, they are doing well. But when we took in outside, say UP, Haryana, then Rajasthan, it is not doing well because of this. They are adapted to the climatic condition. But my strong belief is that Saiwal will certainly do better in eastern region of our country. Any other?
Sir, one question from actually it is from me. Actually,
Okay.
I have visited the Kutch region of Gujarat, and during my visit, I have seen that two most important breed there. One is Bunny Buffalo, and another is Concrete. And I have seen that this Kutch region, there is no means provision of even water or means green fodder. Even those Bunny Buffalo is giving more than 20 liters of milk per day. And even and Concrete cattle, I have seen they are highly adapted in this Kutch region. Okay, so can this breed can be adapted in this condition? There is hot, dry condition. Some of the districts like Purulia, Bankura districts of West Bengal or Birbhum district. That is
Okay.
only buffalo. Or
Okay. First, I will come to Bunny Buffalo. If you see that geography, no, the Punjab, Gujarat, and Pakistan, it is not a far away place. So Bunny, it is now established breed, but it is an extension of Kundi. Kundi is a breed in Pakistan. Okay. So
Yes.
Kundi and Murra is nothing but a single population. So in India, we call it Murra, means the horn is carved. The same thing in Pakistan, they call it Kundi. So this is an offshoot, or say this is a animals of this group, Kundi or Mura, whatever you say. It was isolated to Bunny grassland. Again, I am telling Bunny grassland. What you saw Kutch area where visitor go, I also visited those places. Those are this run. Run is a different area, but Bunny is a grassland. So they survive on grass. Nothing was fed to these animals, only salt and other thing. Yes, but grass is required, and your humidity is also required. Oil, this water is also required. Grassland is a marshy grassland. If you have gone to Bunny grassland, it is a different story, not Purulia Bankura. So it is as good as you can equate with a Murra buffalo. If Murra is doing well in West Bengal, Bunny will also do. Regarding Concrete, we are not knowing Concrete is so beautiful animal or breed. Earlier, Concrete was considered as a draft breed for draft purposes. But subsequently, we find there are good yielder, even if 4,000 liter per lactation animals were available. So government of India or say ICR certainly, the Concrete is also taking initiative to improve this breed through selecting superior sire. We call it progeny testing program to take this Concrete. Certainly, Concrete may do better under this any condition, the harsh condition in any parts of the country. Concrete, then Rati also can do well. Any other?
Okay, sir. One question from the participant. That
Yes.
presently, mostly pedigree selection of bulls being practiced. Are we misleading farmers producing cement doses based on those dams yield?
Okay. If you have followed my deliberation, I told science has gone a far way. Genomic selection, and people possibly after 5-10 years they will say metabolism selection, not even genome, proteome selection methodology. So, but in India or in most of the developing countries, we are still in the conventional stage, conventional selection strategy. What is conventional selection strategy? Nothing. That is nothing but, in case of milk yield, it is a protein testing program. Okay. In the protein testing program, sire does not give milk. So to assess the sire's ability, we allow them to produce daughters, and daughters' milk record we see. Based on daughters' record, we evaluate sire. This process is called protein testing program. Now, in a protein testing program, we put 15 to 20 bulls. So whose 15 male calf will be the bulls, future bull? Based on the dam's performance. That is possibly you are talking pedigree selection. Initially, when we are putting the test bull in a protein testing program, based on the some criteria has to be there. No, that criteria is dam yield. If it is better, we take those, say, 30 male calves. 30 male calves were reared. We see the semen quality, semen attributes. If they are giving good semen or semen good quality, then out of that we ultimately select 15 test bull. We allow them protein testing and come up with, out of 15, 2 bull are proven bull. They are actually colored bull, or say with a feathered bull. We use them to improve the population. So pedigree evaluation and the process, if you see, the test bull we are using in the field condition, the animal or the bull which will not pass the test, means 2 we are selecting, 13 will not be there. We will not select. So those 13 will also produce progeny, but that progeny will be certainly superior than the average population. Best is the 2 bulls. Will be evaluating protein tested bulls will be the best one. The second best one will be remaining 13, which were selected in the calf food based on his dam yield. So it is no harm. It is better than doing nothing, So that way, pedigree selection, we are not stopping there. Pedigree selection, first stage, second stage is protein testings. Okay. Clear?
Sir, actually, I have one question. Dr. Gospel telling.
Yes.
Sir, can I put
Yes,
you on
sir.
question? Yeah.
Yes, certainly.
Okay. Actually, sir, there is one technology, that is a CRISPR technology is coming in very fast way. So whether it can be used to introduce specific climate-friendly gene editing for the development of climate-smart breed.
Okay, you are technically correct. Theoretically, answer is yes. But breeder always we see the population, not 1, 2, 3, 4, 5, or 100 or 500. When we are talking about millions of. I told that in my first answer, non-descriptive cattle, it is 146 million. Means 14 crore or 15 crore we are talking about. If we can produce, say, 10 crore of this sort of animals by CRISPR technology, certainly it is a huge thing we have done. But practically, possibly, it is not there. As a what is that? You can demonstrate in few animals, but that is not a breeding strategy. You cannot use them. We'll have to see those variation is heritable or not to establish that. If it is not heritable, then it is useless. Okay. So a lot of research is required. We have to see whether it is heritable or not. If it is not heritable, that changes is meaningless for a breeder.
Oh, thank you, sir.
Thank you very much. Thank you, the rapporteur, Dr. Mukherjee, Dr. Banerjee, Dr. Bose also, for your nice presence of these questions. I again thank Dr. Dev for your nice deliberation and beautiful discussions to clear the different questions from the participants. I think it is all of the participants now clear what type of problems we are facing and what maybe the our coming future research areas of genetics breeding point of view to upgrade the quality of our indigenous livestock, and particularly in this climate stress condition. So again, Dr. Dev, we are very much thankful to you for your past presence and your beautiful discussions. Now we are going to our next program, the Valedictory Program. Our Dr. Dev is the chief guest of our Valedictory Program. We are now already completed our 7 scientific sessions, and at the end of this Valedictory Program, we will join in national anthem. So I am requesting to all of you, please stay with us up to the end of the Valedictory Program. Before the starting of this Valedictory Program, there is one important announcement regarding the certificate of the participants. It was declared that the presence of your first, second, and third day, the feedback of the second and third day is important, and that participants will get the mail in their personal email, one final feedback form. You please fill the form in a very cautiously, because any spelling mistake if you done there, then there will be reflected in your auto-generated certificate. And there is a space scarcity also. So you follow the character space which was mentioned in the form. Accordingly, you put the all the informations there so that your certificate will be fulfilled. And you please use the code number because your identity is recorded in our system through the code number only. So if you do not write the code number properly, so there will be a problem to send the certificate and other feedback forms also in your mail. So I think all of you enjoyed very much. Now I am requesting Dr. P.R. Ghosh, Associate Professor of our Physiology Department, to moderate the Valedictory Program. Dr. Ghosh, now please start.
Thank you, Professor Das, for giving me the opportunity to moderate this session. First of all, I want to convey my good afternoon and best regards to all the honorable dignitaries, speakers, and dear participants. Now I am happy to announce in the valedictory session we are having with us our Honorable Dr. S.M. Dev, Sir, Head of Animal Genetics and Breeding and former Director of NRC IAC. Sir, I am requesting you to be the chief guest. It's a great pleasure to invite Professor Subhasis Bottabell, Dean of Students' Welfare, Head of the Department, and Professor in Veterinary Biochemistry of our university, to be the guest, as a special guest. And whether Professor, our Dean is there?
No, yet not joined.
Huh? Yet not joined. Okay. Okay. When he will join, then we will embrace him. And also we are having with us our very respectable teacher, Professor Sagar Sannal Sir, our former head and professor of our department, and I am requesting him to be our special guest. And we are also with us the stalwart speakers of the 3 days workshop, Honorable Professor M.L. Madan Sir, Dr. D.N. Kamra Sir, Dr. Vikas Das Sir, Dr. V. Sejian Sir, and the others. And respected Dr. P.K. Das, Head and Program Coordinator of the workshop and his team, respected enthusiastic participants across our country and abroad, ladies and gentlemen. I am feeling humble to each of the dignitaries for their illuminating presence in spite of their busy schedule. I heartily welcome all you, sir. Let me call it fraternity of scientific minds here together, and on my own behalf, extend a very hearty welcome to all speakers and participants for gracing your important talks, opinions, and excellent deliberations since last 3 days. It is high time that such an event took place to highlight the remarkable importance of climate-smart livestock production in these difficult days. I hope the participants had 3 most productive days of interesting and stimulating discussions. I sincerely believe that this workshop will be a great success, not only as a chance to share knowledge and experience in the subject, but also as the beginning of a long and fruitful cooperation and friendship among fellow participants and the speakers. We have tried our level best to make you concerned and updated through this workshop. The excellent talks in all the sessions made our task easy. We are really grateful that such colossal personalities like Professor Madan sir, Dr. Kamra sir, Dr. Sejian sir, and many more not only delivered talks but followed other lectures with great enthusiasm. Sir, to us, you are not only the river speakers. We perceive you become the most valuable team members of our organizers. Thank you, sir. I adore your greatness. Dear participants, climate change will eclipse to our silent livestock production, and after this workshop, we certainly got some ray of hopes. Sir, I am humbly submitting to you all, though Department of Veterinary Physiology is not exactly a Spartan child, but it had acquired many Spartan habits of disciplines and togetherness. We have limited resources, but we possess the zeal to outperform. We are thankful that your undented support and inspiration instill confidence and passion to us. May the excitement and fellowship that you shared create happy memories and smiling magical moments. Once again, I would like to extend a warm welcome to you, one and all. Thank you very much. Now, I am requesting Professor Pradeep Kumar Das, Head Department of Veterinary Physiology and Program Coordinator of this workshop, to place the synopsis report of the workshop. I must mention his leadership and continuous motivation to lead ourselves for taking this venture. A man behind in true sense. Professor Das, please.
Thank you, Dr. Bose. Respected Dr. S.M. Dev, Chief Guest of the program, Dr. Miracle Bose, Guest of Honor, the Dean of our faculty, Dr. Subhasis Bottabel, PSW and Head of the Biology Department, Special Guest, Professor Sagar Shangal, Sir, Special Guest of the program, our eminent speaker, Dr. Kamra Sir, Dr. Sejian is here, and Dr. Khan is also here, and all the teachers of the department, the teachers of the university, the officers of the university, my dear students, colleagues, and staff of the university, and the participants. We, on behalf of the Department of Veterinary Physiology of West Bengal University of Animal and Fishery Sciences, plan to discuss the various researches and experimentation that are continuing to enhance the resilience in climate-smart livestock production scenario from the expert of multidiscipline, considering the significance of livestock in GDP and livelihood in India. As such, we organized the 3 days online national workshop in climate-smart livestock production in India. The workshop was inaugurated on 5th August by our Honorable Vice Chancellor of West Bengal University of Animal and Fishery Sciences, Professor Chanchal Dubey, where eminent physiologist Professor M.L. Madan in the spirit of the workshop as chief guest. The EREF of the university, Professor A. Goswami, and the Dean of the Faculty of Veterinary Animal Sciences, Professor N. Ghosh, were present as special guests and enlightened the impact of the climate changes in livestock production. Entire discussion was conducted in several scientific sessions. In first session, Professor M.L. Madan, distinguished physiologist, in his excellent speech on challenges in climate-smart livestock production in India, entailed the innovations to forecast and impact climate changes and physiological system approach in climate-smart livestock production. He nicely explained in detail to achieve such goals of food and nutrition security, adaptation, and mitigation. He elaborately described and urged the necessity of longitudinal profiling studies at body, system, organ, and at the molecular level. He highlighted the holistic approach and vision to all of us in PSLS production. In second session, Dr. Veerasamy Sejian, Senior Scientist, NINT Bangalore, in his gem of lecture, enriched the latest development of climate change research not only in small ruminant production but also on climate system and global climate change and its consequences. He smoothly traveled through about all the aspects of the climate change like heat stress, heat impact, nutrition and working stress, concept of multiple stresses, emphasizing infrared thermal imaging. The beauty of his lecture is that it's mainly based on of his own research work in collaboration with global leaders. He nicely put forth the future perspective. On second day, in third session, Professor D.N. Kamra, ICAR National Professor, Division of Animal Nutrition, ICAR IVRI, was concentrated his discussion on greenhouse gas emission from livestock and their mitigation, emphasizing the different greenhouse gas production from anthropogenic sources, including animal agriculture, contribution of livestock production to the total greenhouse budget in different countries, and ruminal methogenic communities. He explained the outcomes of several experiments in respect to the role of phytochemicals, various feed additives, plant extracts, and methane analog, which proved that such intervention not only reduces the methane emission, also increases the growth and production of the livestock by increasing the feed efficiency. Professor Kamra suggested for large-scale research in vivo studies emphasizing the role of gut microbiome, phytochemicals, and dephonation in the perspective of climate-smart livestock production. In discussion, he clearly stated that culmination of unproductive animals would be one of the major ways to mitigate the issue of methane emission from the livestock. In fourth session, Professor L.K. Babu, Dean College of Veterinary Science and Animal Husbandry of OUAP, explained the new approaches to climate-resilient livestock production as an expert from livestock production management. He comprehensively discussed different regions of India with greater heat stress to the livestock and different climate-resilient animal technologies such as breeding and nutrition, thermotolerance in different genetic breeds, and designing of livestock sets in different agroclimatic zones to combat the heat stress condition in India. In fifth session, Dr. D. Banerjee, Assistant Professor of Department of Veterinary Physiology of WVFS, confined his talk on impact of climate on livestock and poultry production in Sundarban. He described gradual changes of the nature of Sundarban due to alteration of climate caused by global warming. At the same time, Dr. Banerjee explained the adaptability of the livestock and poultry in Sundarban areas in changed climate, emphasizing their potentiality and threats. He pointed out various limitations of the livestock and poultry productions like lack of knowledge of scientific rearing systems, scarcity of input like quality breeding stock, beef, fodder, vaccine, AI, etc., and market and milk poultry products. On third day, in sixth session, the lecture of Dr. Bishi Das, Principal Scientist of ICAR NRC of P. Guwahati, on impact of heat stress on livestock production and health, enlightened the physiology of stress and about the thermoregulatory profiles of animals as well as the heat exchange mechanism between the animals and environment. He discussed different assessment tools to evaluate heat stress in animals. His lecture reviewed the alteration in physical, biochemical, metabolic, endocrine, and reproductive function upon the heat stress. The information obtained from his talk will help to formulate appropriate management and nutritional strategies for sustainable animal production. In seventh session, the discussion on the topic genetic adaptability and resilient livestock production under environmental stress by Dr. Asian Lake, Head Division of Animal Genetics and Breeding of ICAR NDRA Karna, encompassed the genetic factors that made the animal long-term or generational resilience to climate change. The candidate genes associated with climate-resilient traits were highlighted for better understanding of inherent genetic makeup of indigenous animals under changing climate scenario. The lecture of Dr. Dev will be useful for marker-assisted selection of highly adapted animals for climate-smart animal production system. All the sessions become attractive during discussion on the points as raised by the participants and placed by their advocates. Dr. P.R. Ghosh, Associate Professor of the Department of Veterinary Physiology, and Dr. A. Patro, Assistant Professor, Department of Animal Nutrition, acted as rapporteur in first two sessions. Dr. K. Das, Assistant Professor, Department of Veterinary Physiology, and Dr. A. Patro, Assistant Professor of Department of Animal Nutrition, performed the function of rapporteur in third, fourth, and fifth session. Dr. J. Mukherjee and Dr. B. Banerjee, both the Assistant Professor of the Department of Veterinary Physiology, executed the role of rapporteur in sixth and seventh session. Entire Google Meet program was streaming by Mrs. A. Dev, CEO, Biotech Hub, VRA of the video. The gem of the workshop are the participants, who are diversified in nature. Nearly 800 participants registered their name for attending the workshop, including abroad, of which 30% was female. The selected participants were of various stratums, starting from students, research scholars to faculties, scientists, subject specialists, teachers of the various colleges and schools, and professionals. 20 is the lowest age and 70 is the maximum age of the participants. The success of the workshop solely depends on the magnitude and acceptance and assimilation of erudic talks by the previous participants. Thanks to all. Thank you.
Thank you, sir, for your brief report of the workshop. And you nicely presented the total workshop in a nutshell. Now, I am requesting Dr. Sitangshu Mohan Dev, the chief guest of this session, and a very close friend of mine, but in a personal level. Actually, he's my batchmate. So I am elated to invite you to deliver your speech and share your valuable thoughts with us. So, Dr. Dev, please.
Thank you, Professor Ruminal Ghosh. Again, my different role. Good afternoon, very good afternoon to all. And I'm really surprised to see, very happy to see that 800 participants are there in this web workshop. So I start with my esteemed senior Professor Sagar Sannal, then Dean, Faculty of Veterinary and Animal Science, Professor Nilotpal Ghosh, Dean of Students Welfare, Professor Shubhashish Bhattabbal, then Dr. D.N. Kamra, my senior, Dr. J.R. Khan, Dr. Sejian B. Sejian, and this program coordinator for this, Professor Pradeep Kumar Das, my friend Prabal Ghosh, Dr. Vikas Das, and all the participants here. If I miss somebody, I apologize. Ladies and gentlemen, I am very thankful and happy to the organizers because they have given me opportunity to speak in such a very beautifully organized program. And I congratulate the whole team and specifically Professor Pradeep Kumar Das for taking this initiative. I understand this is a very meticulous planning and insistence and putting all the people together to make this program is successful. It is a very difficult task, especially in the situation where this COVID-19 has done a havoc to us. I really thankful and happy to see this has been, it has taken a very good shape. So friends, climate smart livestock production in India, this workshop is, I could not attain all this, but certainly I was watching or say involved in some piecemeal manner. The eminent personalities like Professor Madan, Dr. D.N. Kamra, Professor L.K. Babu, Dr. B.C. Das, Dr. Sejian, Dr. Deepak Banerjee, during these 3 days, and I'm quite sure that we are all have a better understanding about this important contemporary issue, that is climate smart livestock production system. We now know better what is it. I don't say we know the best, but what we are knowing 3 days back, we certainly, all the participants knows better what it is, how serious the climate stress is, and how it is affecting the physiology of livestock, and finally, the livestock production and productivity. If I understand properly, the climate smart livestock production system revolves around 2 things. Possibly it has been explained. One is mitigation of greenhouse gases, means livestock is contributing to the creation of climatic stress, or say global warming, and that implies the climatic stress, again coming back to livestock as well as human and all living organisms. And second aspect is adaptation of this livestock, certainly all biological system, to this climatic change or climatic stress conditions. So these two are the pillars, I would say, through which this climate smart livestock production system revolves around. If I say climate change, it is not the only reason or constraint contributing to the hindrance to the livestock production system. There are other players also, but in this workshop we have discussed on this climate smart livestock production system only. So to mention some, suppose population density, human population density is another factor, income of the people as well as coming from the livestock production system, then diet shift of the people, because consumerism is increasing, so we require more protein, more milk, more egg. So there is a huge demand, and the demand in the developing countries is far higher than what is the demand in the coming days in the developed nations. Then public perception about this livestock production system and resource constraints, certainly a biggest factor. And this climatic stress is the issue, burning issue we have discussed here. The climate smart livestock production system may be achieved. If I say smart means how best we can use the livestock production system under the climatic change scenario. There are different technologies available with us, possibly during this 3 days deliberation. We have seen a lot of technologies available, how to reduce the methane, how to imbibe this, more adaptability in this animal. There are a lot of technology which can be classified into animal and farm management, including shelter and waste management, animal health management, supplementary feeding management, vaccination against rumen methanogenic flora is also technology available with us. Then grazing and pasture management, possibly we have not discussed, but that is there elsewhere. And in our country, it is a hill and other some of the areas where this pasture and grazing management is also equally important. And creating warning system and insurance, we have the technology with us. Agroforestry is another technology with us. Breeding management certainly is another technology with us. We have certain technology available with us. So we can use this technology which are available in some of the areas. In some of the areas, these scientists, academicians, they are working hard to develop technologies, because these are the truth we'll have to face. Nobody can wish away that tomorrow there will be comfortable climate. It is going to increase. If you see the trend world over, see in 100 years, there is an increase in 1 degree centigrade temperature. So this will continue. Maybe in the future days, the demand of food and this different food quality protein food will be increasing. So we'll have to make a balance out of this. So increasing awareness among stakeholders is very much required. Modification of technology for simplicity and economic. This is also one of the important. There may be some technology, which is a high technology, costing a lot to the farmer, a lot of money to the farmer. possibly adoption rate will be very low. We'll have to develop newer technology to deal with this different stressful condition, and needs policy intervention from the government also. Government to understand, unless if we keep the farmers to the vagaries of this environmental stress, possibly they cannot sustain. And the animal who will survive may not be economical, they may not produce more. And they will produce more greenhouse gases and will be unnecessarily blamed. We have no choice. We'll have to make a balance out of this thing. But one unfortunate thing is that we don't understand how farmers appreciate or perceive this climate change scenario, and how other stakeholder takes the decision, whether they are aware of it. When we call it environmental stress or say climatic stress, it is something related to industry only. It's not that it is everywhere. All the components in the production system, it is a livestock production or industrial production or any production system, we are actually putting pressure on our environment, and environment is punishing us. We'll have to look those things into consideration. We'll have to understand the economy of the livestock production system, incentives to the farmers, and the technologies required to be developed. And possibly we need to understand a lot in this area. This is a beginning, and I hope that this sort of deliberation will make a opening for other places to discuss and come up with technologies for better climate smart production system to be in place in our country. With this word, I thank again. I am very happy, and my best wishes for this this workshop, and thank you.
Thank you, sir, for your precious remarks. Actually, you delineated the theme and the concept of our workshops in a nice way. So I think your suggestions, your thoughts will be certainly be an asset to us. Thank you again, sir. Now, I'm happy to announce that we are having with us Professor Nilotpal Ghosh, the Dean, Faculty of Veterinary Animal Sciences, West Bengal University of Animal and Fishery Sciences. I welcome you, sir. Professor Ghosh, I welcome you, sir. And I'm inviting you as a guest of honor. And sir, now it is your turn to deliver address to the all the participants and the speakers, dignitaries. Thank you, sir.
So good afternoon, everybody. We are nearly at the end of this 3 days workshop organized by our Department of Veterinary Physiology. Actually, I am feeling happy to be in this platform with chief guest of this session, and Dr. S.M. Dev. I'm very happy to see you, sir, after a long period. You are the senior of our batch in this university. And along with him, I'm also feeling happy to announce the name of Professor Sannal, my teacher, as special guest of this program, and also our Dean Students Welfare, my beloved Dr. Botokgal, and some senior faculty members from all over the country is also present in this session, and Dr. Kamra is there. What I should say that we are really happy to have renowned and eminent scientists in this program, rightly from veteran scientist Dr. Professor M.L. Madan to our young scientist Dr. Deepak Banerjee of our faculty. So there is a well blend from veteran to young scientist with this in this platform. Actually, Dr. P.K. Das, Pradeep Kumar Das, the organizing secretary of this program, nicely pointed out in this session that what we have discussed during these last 3 days and Dr. Dev also pointed out some other areas where we should give some emphasis. He rightly pointed out some aspects, but the topics, what I feel that were nicely selected, where the effect of climate on the livestock production were focused, discussed by Professor Madan, Dr. Deepak Banerjee. And another aspect is the contribution of greenhouse gas from livestock sector to the global warming cause is also there, rightly and nicely presented by Professor National Professor Dr. Kamra. So both the sides were blended in this program. It is true that some other aspects, as Dr. Dev pointed out, should be considered in broad sense for a better livestock production, and the livestock production to be very smart. All these aspects should be considered. Otherwise, there may be some problem or lacunae. Actually, what I have heard that more than 800 participants have attended this program, and generally, in case of conventional seminar, such number is not generally seen. So this is an opportunity that many people are gathered in this platform, and hopefully they have enjoyed this program and gathered some experience. And Professor Dev rightly pointed out that awareness generation is very important. We have many technology with us. Here also, the techniques were discussed, how to mitigate the effect of climate, as well as how to reduce the greenhouse gas from livestock sector. Techniques are there, techniques are validated, it is with our hand. But we have to carry this technique to the farmers level where the livestock production is in operation. So these participants should be the bearer or the connector between this scientist forum and to the livestock farmers forum. And if they are benefited out of this program, and if they carry these techniques from this laboratory, so-called laboratory, what we have discussed in this platform, to the farmers level, so that our livestock to be smart, then only we consider such type of program successful. With these few words, again, on behalf of the Faculty of Veterinary Animal Sciences of West Bengal University of Animal and Fishery Sciences, I congratulate you all, because we have the panel of experts, we have the participants, without which the organizers cannot do anything. So I congratulate all of you. I thank you again, and stay safe. Thank you all.
Thank you, sir, for your excellent support and remarks in the concluding session, rather say the valedictory session. Your suggestions and all these things will be our future guide. Thank you, Professor Ghosh. Now, I'm elated to invite our respectable teacher, Professor Sagar Sannal sir, former head and professor of the Department of Veterinary Physiology, that is our department, and special guest of this session. He's a great teacher and motivating mentors to a lot of us. We are eager to hear your excellent thoughts, sir. Sannal sir, please.
Good afternoon to all. And I want to convey my thanks to Professor Das, P.K. Das, for this, for inviting me in this session of the valedictory session. And I am happy that the Professor Dar and its group has made it a successful webinar seminar, and ever I attended. So I'm also proud to be here among the host of stalwart persons, including Professor Madan, Professor Kamara, Dr. Sejian, Dr. B.C. Das, Dr. Banerjee, Dr. S.M. Dev, Professor J.R. Khan, and Professor Batupal, etc. And there are a lot of host of teachers are here. And I'm a small among the vast experienced persons of this gathering. And I also want to convey my thanks to Pradeep to select the item. That means the topics of this session, that smart climate. Nowadays, in 2020, everything is smart. Mobile phone is smart. Coronavirus is smart. And resources are also smart. So smart climate must be very important discussion point here. Now, Dr. Madan has cited many examples of how the livestock production give the bounce effect on the Indian economy. And he cited the name of the two states, Punjab and Haryana. They are mostly affluent and wealthy section because they are most on the agriculture and livestock farming. But I like to say that not the Assam, not the Haryana or Punjab. This is the importance of geographic location of individual states. So all states are not like Punjab or Haryana, but you can see the Maharashtra or Karnataka or Tamil Nadu or Gujarat, they are shining in the industry and giving impetus to the Indian economy. So this is my observation. And also, there are some poor states where the large mineral deposits are there, but not exploited so much. That is my observation. And regarding the greenhouse gases, Professor Kamara has said that the reduction of methane gases from the large animal is to reduce the consumption of red meat. Sir, I love to eat red meat. Don't ask me to reduce the consumption of red meat. But similarly, I request you, you have formulated some indigenous herbs to reduce the methane production. But I request you, whether you have tested any scientific experiment or any conducted in scientific experiment regarding the conversion of methane to any biomolecules before its emissions. The conversion of methane into any biomolecules before its emission. It will help to reduce the greenhouse effects of the large number of unproductive animals in India. So you have experimented with the different system, and you have shown that the body weight gain. But what I like to say that body weight gain, what about the production? Is there a reduction of milk production or increase of milk production? So you might have some thinking about this. And also, Dr. Sejian asked for any vaccine can be generated. So Sejian, he's my student. I like to apprise you that these methogenic bacteria are not has any antigenicity. So is there no antigenicity, vaccine cannot be produced. And also the high temperature effect, rumen temperature versus liver temperature. And it is very important, the difference between rumen temperature and liver temperature to assess the heat stress also. And Dr. Deepak Banerjee, very young scientist, I love him, he's my also student. He is conducting some experiment on the Sundarban regarding the adaptability of some animals and the harsh climate. So, Dr. Banerjee, you have to go certain few miles to establish the sodium content of the urine, because you have to go to the ratio between sodium and potassium in the blood, in the urine, in the interstitial spaces likewise, and also the genomic regulation of AT1 and AT2 receptors. So this is very important because there is no taste difference or high quantity of sodium in marine fish versus sweetwater fish or brackish water fish. And also you have to study the soil and the fodders or the vegetables used to consume by the goat. Is there any higher level of sodium or not? This is my observation. And my findings or observation would say that if you want to reduce the greenhouse gas effect, we have to eliminate the large number of non-productive animals through the slaughterhouses. It will boost up the deficiency of good quality of protein source. And this is important because the India is one of the largest concentration of the non-productive animals in the world, which has no effect, only emission is the important here. And these are my observations. And another is that we have to select the breeds suitable for a particular geographical locations of the states regarding temperature, humidity, rainfall, etc., for the soil. And last, the BC Das states that trace versus cortisol. Dr. Das, your deliberation is very good in respect of thermal trace, but a cortisol level is very difficult to establish the trace always. The thermal not only trace, which trace you don't know, whether it may be internal or anything else, the cortisol level varies frequently, so it is difficult. And Dr. Dev has suggested that selection, this breeding type of breeding selection or the selection of the progeny, actually selection of the sire and dam from the genetic point of view. It is very important, but I don't know how long it will take to evolve this process in regular basis in our country. I am thankful to all the listeners and all the scientists and stalwart persons here, and I like to convey my best wishes to all the participants throughout the India as well as the other countries. And finally, I convey to Dr. P.K. Das for the elaborate arrangement of this seminar. Thank you all.
Thank you, sir. Sagar Sannal, sir, for your excellent remarks and explanation. You are always dear to us, and your blessings, we hope that your blessings will always shower on us forever. Thank you, sir, again. Now I am inviting Professor Subhashish Bottombell, Professor and Head, Department of Veterinary Biochemistry, and Dean Students Welfare, and the Dean Students Welfare of our university to deliver his address. We are eager to hear you, sir.
Good afternoon. First of all, I must give my thanks to Professor Kumar Das and his entire team for inviting me as a special guest for this session. So here in this session, I must give my regards to my teacher, Dr. Sagar Sannal, my teachers, Dr. D.N. Kamra at IVRI, my senior, Dr. Sitanshu Mohan Dev, now that former director, NRC Diran, Dr. Nilotpal Ghosh, Dean, Faculty of Veterinary Animal Sciences, WBFS. And I was present in the inaugural session. I must give thanks to the inaugurator, Professor Chanchal Untitled Chapter, sir, our Honorable Vice Chancellor, and the Professor Madan sir, the eminent scientist and the former the DGD, and all the speakers of different sessions, Dr. Sijian, Dr. Vikas Chandra Das, like Professor Babu, L.K. Babu, the Dean OUAT, Dr. Deepak Banerjee, and all the participants. I must give thanks to Professor Pradeep Das, the Professor of the Department of Physiology, the organizer, and his team for choosing this topic, which is most appropriate in the present scenario. Regarding climatology, actually, when I was student in IVR, I have given that one division, the physiology and climatology. On my interest, I was there in the experimental climatology, that lab. The then scientist, Dr. Joshi, actually he was the eminent physiologist, and Dr. Tripathi, they were there along with some other scientists. So I have seen first time how experimentally that different climate have been produced, and the production and physiological studies and other studies have been by different scientists. So this is very appropriate topic, and Dr. Das had designed in such a way that so many aspects have been covered. Dr. Omlan Patto was there, one speaker. So I want to give my regards to all my seniors, including Dr. Dev and my colleagues, Dr. Das, Dr. Sijian. They have nicely narrated all the importance of different aspects. So all the areas have been covered. So on the behalf of students' welfare community of India, I am thankful to the organizer for conducting such type of this web seminar on the climate smart livestock production in India. Actually, from different speakers, we learned that so many techniques have been developed. Dr. Dev says, and different scientists, that various speaker says, that different technology have been developed. And we have to adopt all this technology and have to apply actually all this technology for our better production. So if we improve the condition in different adverse condition and adverse climatology, so we are able to improve the production in different levels. So our students have been benefited. I have heard that more than 800 participants. So from different sectors, some are students, some are research workers, some are scientists, some are teachers. So I must give thanks to Dr. J.R. Khan, he's present here, the eminent physiologist from Dug Veterinary College, and other scientists who have been participated in this 3 days seminar. I must give thanks to our university authority for allowing us for conducting such type of workshop. So I will request Professor Das and his team to conduct such type of seminar in near future also, and we must give our message to the future workers and farmers as well. So then only our objective of this seminar will be fulfilled. And I must give thanks to Professor P.K. Das, Dr. Pobal Ghosh, Dr. Kingsuk Das, Dr. Jaideep Mukherjee, and Dr. Deepak Banerjee, the entire team of Physiology Department, and Ms. Day from our DRF section for nicely conducting this very in worst situation of COVID pandemic, this nice seminar they have conducted, and our students have been benefited from this seminar. Thank you once again to the organizer for giving me the opportunity as special guest in this session. Thank you all.
Oh, thank you, sir. Thank you, Professor Botobel, for your valuable and enriching address. Actually, the physiology and biochemistry are always moving, holding hand by sides. Okay, so it's anything which we gain that is we are proud for you. I always think in this way. So, and we believe that your suggestion and support will guide us to move forward now and also in future also. Now, I am humbly requesting Dr. D.N. Kamra, sir, pioneer in this field, to deliver his comments on this workshop. Dr. Kamra, sir, please.
Thank you very much, sir. You are able to listen to me?
Yes, sir. Yes, sir. It is nice. It is clear and loud.
Yeah. It was a really very impressive and very nice arrangement for this webinar, which we have attended for the first time. Before that, I attended only one meeting on webinar, means on web, of the Research Advisory Committee of CIRC Merit. And it is the first time that I am listening to so many people, and we are meeting so many stalwarts of animal nutrition, physiology, and biochemistry, and other subjects that we are able to see each other, at least see each other, not to meet, but actually seeing is sufficient. And under these conditions, in coronavirus is creating trouble for all of us. The 7 topics they have identified, they are really very impressive. None of the lectures was duplicating each other. That is a very good thing, because in a very short time, 6 hours of in 3 days, 7 lectures on different topics, and some of them was really very impressive to me also, like genetics by Dr. Dev, because we know only the surface knowledge of this genetics and genetic improvement of microbes and animals. And he has made it very in simple language and very elaborative that this is one of the way we can have the stress of the animal, stress of heat or other stressors on the animal. So therefore, I really congratulate all the organizers of this webinar who have taken a lot of time, lot of information they have given. They have sent this email to maybe thousands of people, and out of which more than 800 people are attending this webinar. And normal seminars, workshops, it never goes to beyond 100, maybe 50, sometimes it is only 20. And now it is 800. 800 is a very big number. So therefore, I congratulate Dr. Pradeep Das, who has been really active force behind this webinar. And I once again thank him, as well as Dr. Patra and all of you, that you have given me a chance to attend this workshop. I really liked it. I attended all the 7 lectures, complete, because they were giving new information, which even I did not have in my 40 years of service in ICR. So therefore, I once again thank to all of you and say goodbye to everybody. Thank you.
Thank you, sir. Hearing you all is dear to us, and every time it generates new impulse to us. Actually, sir, from the first day you are there, and actually your the thirst for the newer things or your perseverance is a really example for us. We'll try to follow you, sir. and I think all the participants are very much thankful to you. They are for your excellent, not only in the party your talk, but your all the times your small suggestions and these things during the different sessions. Thank you, Professor Sir. Now, I am requesting. There is another speaker is there who didn't spoke today, but ultimately Dr. B. Sajian. He's a student and is a very young and already he got a lot of accolades on his crown. So I am requesting Dr. Sajian to share your thoughts in a very brief manner. Dr. Sajian, please.
Thank you, sir. It's been a pleasure as always, because when Pradeep Da is organizing something,
thank you. Thank you, Sajian.
So you can expect the things to be.
Your sound is not coming. Your sound is not coming. Sajian, your sound is not coming.
Queries. That got in genetics and breeding, not very many people are working on this aspect. And I'm so impressed with your lectures today, because it got so much of information and the way forward for breeding policy especially. So like as always, means Kamra sir is our guru type of a figure, and he always helps us, with his very modesty, and also we as an youngster very important, very remarkable number in a given Indian scenario where we are very new to this type of technology, online lectures and other things. I'm so impressed with the way the things have gone, especially from the Webs College and especially Deepak Banerjee and other people who wanted to work in collaboration with us. We'll be very happy to associate with these people also, and also some of the participants who want some sort of guidance. We are always open, like Kamra sir did for all of us, and we are also here to take the baton and run forward, following in the footsteps of these people. And thank you so much for the opportunity to be a part of Pradeep Da, as well as it's been a pleasure to see you after a long time. I'm so happy and delighted to be a part of this small program. Thank you so much for the opportunity.
Oh, thank you, Sajian. Thank you, Sajian, for your excellent talk. Our best wishes for you, and thank you. Now I'm requesting Dr. Bishidash to speak in a brief about in the valedictory session. Again, Dr. Das.
I don't know either.
Dr. Das, your sound is not coming.
Hello, can you hear me now?
Yeah, you can hear me. We can hear.
Okay, so this is, I really find it a great pleasure being a part of this webinar. In fact, when Dr. Das, PK Das is the organizer, means he won't leave you any moment until you are ready to deliver your speech. So every time he will be asking that whether PowerPoint is ready, these, so many things. And of course, I had no choice, but he forced me that this is the topic you have to speak about. And I readily agreed and worked with my things, because when we work in a system wherein I just keep on moving from institute to institute, Being at IVRA, in the main campus, I had a scope to work on stem cells. I worked there for 7 to 8 years. Then, while I was shifted to Calcutta Center, I had no scope to work in that field. Obviously, I switched over to stress physiology. And now I am at PIG. Here also, I thought to focus on the stress physiology. And with that idea, very recently, I have submitted a project with the piglet winning and its stress to DST SERB. So first phase of the screening is over. Now it is with the expert for the next reviewing. So I'm hopeful to have this project from DST. Once it is through, again, I'll be working on this stress physiology. And then, of course, being in a webinar when Dr. Kamra, like personality, is there, it is always a real pleasure to see him and his nice talks and the kind of encouragement he gives every time. What I have seen at IVRE main campus, it is really gives us confidence when Kamra sir is coming or when he is there in the seminar. And I have seen his patience hearing all the time throughout the seminar, whole 3 days he was attending. And likewise, when he is there, you will try to speak the substance. I also felt happy seeing Sagar Babu and how nice of a gentleman he is as a teacher. Really, I won't forget his contributions, what he has infused to us. And I could see my as a fellow friend and junior colleagues at the college, Dr. Sejian, and after a long time. And of course, Dr. Dev, his presentation was covering so many aspects of the genetic types and its related the stress responses. It is fantastic to hear Dr. Dev just about his topics. And Dr. Madan, probably right now he is not there. His presence also motivates us all the time. And I could see J.R. Khan sir also he gives patience hearing. Right now also he is there. Namaste sir. And likewise, Dr. Nilat Paul Ghosh, Dean sir, thank you very much. Dr. Watopbal, thank you very much. And also my sincere thanks to Dr. Deepak and Dr. Jaideep and all others there in the Dr. Pikeda's team. My sincere thanks to all of you for conducting such a nice webinar in a very fantastic manner. And probably this is the COVID pandemic situations which could allow so many participants to be in this seminar. And it is remarkable that more than 800 participants, what probably we have never seen in this kind of seminar when we have it physically. I never seen such a kind of attendance. It is mountainous. And of course, it is the persuasions of Dr. P.K. Das. I am sure that probably because of his personal attraction, many people just thronged into this program. And at the end, thank you very much, Dr. Das, for giving me a scope to speak on a pertinent topics in this webinar. Thank you very much.
Thank you, Dr. Das. Your talk are always refreshing to us. Thank you very much. Now I am requesting Dr. Javed Khan, our very noted physiologist, annual physiologist of India, now working as a professor in Durg. And Dr. Khan, could you hear me? Dr. Javed Khan? Javed sir? Dr. J.R. Khan?
Dr. Khan, please unmute.
Please unmute and comment.
Yes. Now,
another video. Okay, thank you.
Yeah.
Thank you, sir. Okay, good afternoon. Yes, thank
Good
you.
afternoon. On the behalf of the participant, I am thankful to the whole team of Dr. Das. You have smartly conducted the SMART workshop on climate. It was a nice presentation. You have covered nutrition, genetics, management, physiology, and everything. You have covered for the stress and environment. So on the behalf of the participant, because I'm a participant for this workshop, on my behalf and on the behalf of the participant, I am thankful to each and everybody for this nice workshop. So smartly, Dr. Das have conducted this program and all the lectures were excellent. And I think that all the participants which are there were benefited by this. And I hope that in future also, Dr. Das will conduct such workshops My best wishes for the next workshop. Thank you very much.
Thank you, sir. Thank you for your motivations and accolades showered on us. And actually, without you, the participants and your active participations, this workshop will be nowhere. So thank you. Firstly, I think if the winners, then the participants are the winners. Thank you, sir. Now, I am extremely happy to announce that Dr. Kingshuk Das, Assistant Professor of our Department of Veterinary Physiology, West Bengal University of Animal Fishery Science, is going to discuss the major outcomes of this workshop. He is an excellent teacher and competent researcher with innovative ideas and scientific acumen. Now I am requesting Dr. Kingshuk Das to deliver his address. Dr. Das, please.
Thank you, Dr. Ghosh. The outcome and the recommendations of the lecture of the first session by Professor Madan on the topic Challenges in Climate Smart Livestock Production in India. First, number one, the basic physiology behind climate change influences production loss or gain as well as adaptation. Mitigation practices in relation to protect livestock production should be critically explored and available ecosystem policy framework to be implemented. Diversification of livestock using different crops or feed varieties and shifting to mixed crop livestock production system would be the most promising adaptation measure to enable the tolerance of livestock to climate change and to disease for improving efficiency and production. Proper policy framework is to be formulated and implemented for effective adaptation and mitigation measures to address climate change and livestock production. In addition, a comprehensive view of cost, time, and effort are required from the producer needs to be included to the policy framework to maintain sustainable production system. The outcome and recommendation of the speech of Dr. Sejian in the second session on the topic Climate Change and Small Ruminant Production, Current Scenario and Way Forward. Research aiming to improve livestock production under changing climatic conditions should aim to counter multiple abiotic stress simultaneously. Sustainable livestock production in the changing climate scenario must target on improving the production efficiency through strategic intervention. Efforts are needed to identify indigenous climate resilient livestock breeds which have the ability to survive in different agroclimatic zones. In the third session by Professor D.N. Kamra on the topic
We should check the sound. We should check the sound.
the ruminal microbiomes, particularly aricomes, should be better studied to understand the mechanism of methane production that will not hamper the fiber degradation of microbiome. The outcome and recommendation of the lecture in fourth session on the topic New Approaches to Climate Resilient Livestock Production by Professor L.K. Babu was that indigenous genetic livestock resources should be properly exploited based on the different climatic zones, and the proper planning of animal shades and feeding can be performed to reduce climate stress of animal in various agroclimatic zones. The recommendation from the lecture of Dr. Deepak Banerjee in the fifth session on the topic, the impact of climate on livestock and poultry production in Sundarban. Lack of awareness regarding scientific livestock rearing, unavailability of quality feed in the saline zone, problem in livestock product marketing are the major constants in the Sundarban region. Realizing the present situation, perspective of global warming resulting to many natural calamities, the livestock production perhaps is the only answer in respect to the improvement of livelihood of the farmers of Sundarban. Therefore, a comprehensive approach is to be taken to improve the quality of livestock available in this area. In the sixth session by Dr. Vikas Chandra Das on the topic, the impact of heat stress on livestock production and health. The use of stress assessment tools should be applied to evaluate the degree of stress. The constraint imposed by the environmental stressors on physiological, biochemical, endocrine, productive and reproductive parameters may be optimized after suitable microclimate modification. Research for breed-specific difference in heat stress tolerance with special reference to indigenous breed of livestock on identification of genes, coat and skin structure. Creation of awareness among the stakeholders and the policymakers on impact of heat stress and economic losses occurred due to the loss of production and implementation of action plan reduce the carbon footprint of every farm unit, thereby move towards sustainable animal and agriculture. The outcome of the seventh session on the topic genetic adaptability and resilient livestock production under environmental stress by Dr. H.M. Dev. Selection of heat histone modification and transposon intersection together with SNP may be used for the marker assisted selection in the future. So these are the outcomes from different sessions of by different eminent speaker, and thank you very much everyone for attending this seminar. Thank you.
Thank you, Dr. Kingshuk Das, for your brief but clear and loud messages regarding major outcomes of this workshop. So thank you again, Dr. Das. Now, we are nearly at the end of the valedictory session. I'm inviting Dr. Deepak Banerjee, Assistant Professor of our department. I think you all enjoyed his excellent talk at Sundarbans yesterday. Dr. Banerjee, I'm requesting you to deliver the vote of thanks of this workshop on our behalf. Deepak, please.
Thank you, sir. Am I audible, sir?
Yeah, you are absolutely audible, nicely audible. Thank you.
Thank you, sir. Honorable Chief Guest, Guest of Honor, Special Guest, Invited Guests, and eminent speakers, participants, ladies and gentlemen. A very good afternoon to all of you. It's my privilege to have been asked to propose a vote of thanks in the occasion of National Workshop on Climate Smart Livestock Production in India. I, on behalf of Department of Veterinary Physiology, Faculty of Veterinary and Animal Sciences, West Bengal University of Animal and Fishery Sciences, and the entire supporting organization, team, crew, even guests in this online platform, let me call it a fraternity of scientific minds here together. And on my own behalf, extends a very heartily vote of thanks to all the speakers for gracing your important talks and sharing with us your valuable deliberation and opinions since last 3 days. Firstly, I want to convey my best regards and thanks to our Honorable Vice Chancellor, Professor Chanchal Guho, for his excellent address and great support to organize this workshop. A big thank to our heartful gratitude to Honorable Professor M.L. Madan sir, and Dr. D.N. Kamra sir, Dr. S.N. Dev sir, Dr. L.K. Babu sir, Dr. B.C. Das sir, Dr. V. Sejian sir, for your efforts towards your excellent talk on different aspects of climate smart livestock production in India. Sir, all of your illuminating lectures mesmerize us to understand the subject and encourage us to pursue our scientific thoughts in this field. Sir, all your continuous presence, despite of the busy schedule, boosted our spirit to the run of this show. I must mention our deep sense of appreciation and thanks to Professor Arunasis Goswami, Director of Research Extension and Farms, West Bengal University of Animal and Fishery Sciences, Professor Nilokpal Ghosh, Dean, Faculty of Veterinary and Animal Sciences, Professor Subhasis Botopal, Dean, Students Welfare, West Bengal University of Animal and Fishery Sciences, Professor Sagar Sannal, former Professor and Head, Department of Veterinary Physiology, West Bengal University of Animal and Fishery Sciences, for their valuable address and encouraging presence in this workshop. We are thankful for your enthusiastic and all-around support to make this workshop a successful one. I may like to express my sincere thanks to Professor Pradip Kumar Das, Head, Department of Veterinary Physiology and Program Coordinator of this workshop, for giving an excellent coverage and conduction of this workshop. I must mention that you not only demonstrated your excellent skill and knowledge to explain the theme of our workshop, but also your intense passion to organize such beautiful event in the scientific endeavor. Further, I express my sincere thanks to Dr. Prabal Ranjan Ghosh, Dr. Kingshuk Das, Dr. Joydeep Mukherjee, Department of Veterinary Physiology, WAPS, and Dr. Amlan Kumar Patro from Department of Animal Nutrition, WAPS, for their excellent conduction as a reporter activities and different activity in the different season of our workshop, and untiring efforts to make this workshop a successful since its inception. I am grateful to you all, sirs, for your commendable jobs. I also wish to express my gratitude and sincere most thanks to Abhirupa De, DEO, Biotech Kisan Hub, West Bengal University of Animal and Fishery Sciences, Deputy Librarian, WAPS, and all their staff for providing encouragement and for their perfect logistic support and guidance extended to all of us to make this program a grand success. Last but not least, I must mention our deepest sense of gratitude and thanks to all our valuable participants of this 3 days workshop, though through different online platforms, for their presence to encourage us and to encourage all as well. I am also feeling humble to all the dignitaries, including the participants, for their gracious presence in spite of their busy schedule. Thank you, sir. Respected ladies and gentlemen, an event like this cannot happen overnight. The wheels start rolling weeks ago. It requires planning and a bird's eye for the details. We have been fortunate enough to be backed by a team of very motivated and dedicated personnel who know their jobs and their results oriented. Sirs, once again, I want to state that we are
grateful to all of you. We thank you for being with us in this evening. It's been a great pleasure for us. Thank you very much. Thank you.
Thank you. Thank you, Dr. Deepak Banerjee, for your excellent Thanksgiving lecture. I personally, and behalf of all, thanking you for your commendable jobs in different, like as a speaker, as organizers, as rapporteur, and blah, blah, blah. So you are a nice person, nice thing in this way. So from the organizer side, I am thanking you. Okay.
Thank you, sir.
Now, I am requesting Professor P.K. Das, head of our department and program coordinator of this workshop, to conclude this session as well as this workshop. So I am conveying my very good evening to you all. Now I'm telling Professor Das to kindly talk.
Thanks. Thank you, Dr. Bose. Thanks to all the dignitaries, the participants, the teachers, students, staffs, all who are present in this Valedictory Program and this national workshop. We again congratulate and thanks to you for your wholehearted participation. And now only single one announcement that final feedback form will be sent to the email of all participants, and you must be careful to fill in the format because your certificate will be made on the basis of your information which you have given on the feedback form. Now let us join in national anthem with the our program will be end with this national anthem. Please participate in our national anthem.
All are requested to stand. All are requested to stand up.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे